Okay, I think we can get started. Welcome to the FARA Flash Talks. I am Liz Sorani. I'm the Director of Research at FARA. And here with me are the moderators for today's talks, Leslie and David, who will introduce themselves in just a minute. And two of my colleagues, Kelly Madden, who is FARA Patient Engagement Manager, and Miriam Rai, who is the Director of Global Initiatives at FARA. So we call these webinars flash talks because we ask presenters to summarize their research in just 5 minutes and with a single slide, and to explain to us what they're studying and the implication of what they're studying. And we ask them to do it in a simple language that we can all understand, even without a science background. And you'll have the opportunity after each talk to ask questions. And if you want clarification or explanation, type your question in the Q&A box. And try, please, to use the Q&A box and not the chat box if possible. Also, these presentations are recorded, will be posted on YouTube. And I believe that the presentation from actually 3 weeks ago, on May 7th, they have been posted, the recording has been posted already. Today, you will hear from two clinicians, Elisabetta and Roderick. Elisabetta is in Austria and Roderick is in the Netherlands. And two Davids, we just joked about that. One, David Berryman is a grad student at the University of Florida, and David Rufino Ramos is a postdoctoral researcher at Harvard. Elisabetta will talk about iron in FA, and we have evidence that in some tissues, iron seems to accumulate, but this is an area where we're still trying to figure out how much this contributes to the pathology. And Elisabetta's work is trying to answer the question of what happens to iron in FA. How does iron contribute to the tissue and cellular defects? And whether, do FAers have too much iron, too little iron, and what this means for developing therapy, especially therapies that target this specific aspect of FA. Then Roderick will tell us about a project that has been recently funded by FARA. He will tell us about his plan to look at nerves, the peripheral nervous system, in FA patients using ultrasounds and MRI, and measure if there are differences between FAers and healthy controls, and also what happens over time to see if these differences change over time. And he will tell us what he hopes to achieve with this study. Then the two Davids will speak about GAA repeats. You probably all know that all FAers have at least one copy of the frataxin gene that has a hyperexpansion of a small sequence of DNA, the GAA, and this causes gene silencing and low frataxin. So they're both looking at ways to correct this defect, either by shortening or blocking the GAA expansion, and David Berryman will speak to this, and or by removing the repeats altogether. And David Rufino Ramos will talk about this specifically. And they also will tell you about the specific tools that they're using to achieve these goals. So let's get started. I am turning it over to our moderators, Leslie and David. Another David, we did not do this on purpose, I swear. And they'll take it from here, and I will see you at the end. Leslie?
Thank you. Hello, everybody, and welcome to this FARA Flash Talk. My name is Leslie. I'm from Amsterdam, Netherlands. And today I'm the moderator, one of the moderators of this Flash Talk. I was diagnosed with FA in 2011, so that's nearly 40 years ago. Currently, I work as a lawyer for the Dutch government, and I'm also a board member of a Dutch non-governmental foundation for FA. I'm glad to see that there's so much research going on FA. and hopefully there's soon a cure, more cures for FA. So that leads me to the first researcher. Her name is Isabella. No, her name is Elisabetta Indelicato. She's from the Medical University of Innsbruck, and she did research on iron starvation in people with FA. So, Elisabetta, on to you.
Thank you, Leslie, for the kind introduction. I try to share my screen. Can you see my presentation? Can you see my slide?
Yeah.
Yes, that worked. Thank you again, Leslie, and thank you to FARA for the possibility of being here. I'm very happy to be able to present the finding of a study that started some years ago. As Leslie said, I work in Innsbruck, a center for ataxia research, and I started to work with FA actually almost 10 years ago. First, during my clinical PhD, I had a project on FA, then during my residency, and now as a neurologist, I work in the clinics with FA patients and in the research. This research is about iron in FA. As you know, iron is a very important element in our body, but it must be tightly controlled. It cannot be too low, it cannot be too high. And as you might know, early studies show that in FA, iron can accumulate in the mitochondria. The mitochondria are these part of cells that are mostly affected by the disease and that are very important for the energy production. So it was always a question if this iron accumulation in the mitochondria may be deleterious for the cell, but little was known about the iron metabolism in general. And we tried to look at it in a larger study that we performed in 40 patients with FA, 40 adult patients with FA that were recruited at our center. In this patient, we looked at several iron parameters in the blood. We took pretty much blood. And we looked at the iron content in a number of organs that are normally rich in this element, like the liver and the spleen, with a special MRI technique that can allow you to quantify this iron content in this organ. And we were surprised to find out that actually a number of iron parameters in the blood and the iron content, for example, in the liver, were lower in FA patient as compared to healthy controls. And also what we found out is that a number of these iron parameter, for example, also the iron content in the liver, showed an inverse correlation with the length of the GAA repeat, meaning that the longer the GAA repeat, the more severe the genetic of FA, the lower was this iron content. And this result, together with the previous ones, suggests that the disturbance in the iron metabolism in the FA in the body are more complex, and that simply to take away iron is probably not the right therapeutic strategy. And it also shows that FA should react actually normally if you supply iron to the body when you need it. For example, if you have an iron deficiency anemia, which is very common, or if they have a restless leg syndrome, which is also very common and is related to iron deficiency state. I think I am at the end of my presentation, and I'm happy to take questions.
Yeah, thank you for the presentation. There are no questions yet, but maybe I got a question. What is the effect of people lacking of iron?
This is a very good question, Leslie, because as you know, Iman, there are a lot of, for example, women that have iron deficiency or iron deficiency anemia. And in our clinical experience, I mean, it seems to us that this is even slightly more often in FA women. But till now, we have always been afraid to give iron because knowing that there is iron accumulation in the mitochondria, but not knowing the rest, then a clinician were always afraid to do that. As far as I know, there are a number of FA clinicians that do give this substitution when, for example, the hemoglobin level are lower under a certain level, but with caution. Our results suggest that FA people would react as the healthy ones if you give iron when it's needed.
Okay, thank you for the answer. Yeah, we have two questions. One from Mary. Should women who have normal hemoglobin and normal MCV take vitamin C?
If they should take, I mean, it depends on the level of the hemoglobin, and it depends which are the causes of this lower level, of course. But it's pretty easy to measure iron also with routine test. So if everything, all the blood tests show that this low level of hemoglobin are due to also lack of iron, FA women should receive it.
Okay, thank you. Another question from Min. What can adult patients take away from this? Should they have their iron level monitored more frequently to make sure their iron level is within normal limits?
I think that there are two main points. One for the routine, for the daily routine, and one for the future research. What concerns the daily suggests that if people with FA need iron, they should get it from their doctors prescribed, if they need it. If they are normal iron level, if they have a normal blood count, there is no need to give additional one, or better, there is no evidence that that could help. For the future research, this results show that simply taking away iron from the mitochondria probably will not help, because that is maybe a consequence of a shift of the iron that is abnormal. So probably restoring the function of the mitochondria will bring to, how can I say, a normalization of this iron accumulation, rather than something that takes iron out.
Could I ask a question?
Please.
Could you maybe talk a little, and you kind of broke up at the beginning of that last answer. So maybe just to confirm, I think what you were saying is in terms of what can people with FA do with this information about iron right now, the answer is it really depends on if you get a blood test and it shows that you are iron deficient, then you need iron.
Yeah.
So can you talk a little bit about like how is this research different than just like someone has low levels of iron, they need more iron? What is this research is looking at something slightly different than that, correct?
Of course. What is different? First, we showed in general that FA people have lower iron storage comparing to healthy control. That is the first time that something like this has been shown. Somehow this seems to explain also some symptoms that they have. FA people have really often, for example, restless leg syndrome, this bothersome need of moving the leg in the evening on when they go to sleep, that in non-FA people is often related to an iron deficiency state. It somehow makes sense. Then iron deficiency through bleeding or other reason is something that is very common in everybody. So also in FA patient, it can happen. Women that menstruate often have anemia and iron deficiency because of that. And till now, there has always been, worries that you cannot treat this condition in FA patient while you would treat it in people without FA, because we always thought, okay, there is too much iron in the mitochondria, so there is in general too much iron in the body, but that is not like this. And a person with FA that already has a problem with energy, with a performance in the daily life, and then also has iron deficiency anemia is even more, impaired than a person without FA that has the same problem. That for the clinical routine, I mean.
Thank you. I have another question. Is there a difference between men and women in FA?
It's like what was interesting in this study is also that in the healthy controls, I mean, we had approximately half women and half men with FA, and the same in the healthy controls. In the healthy controls, everybody had a normal blood count, women and men. And the FA cohort, there were some women that had lower hemoglobin. So already open sign of an iron deficiency state. So in general, the women in the general population more often have this problem, and I think that FA women have it even more marked. And I don't know, maybe that is also one of the reasons why there are some big studies in which it's shown that Friedreich women are actually slightly worse in the neurological test and the performance test comparing to the men. There is, I think, a big study from the American registry that shows that sex, gender is a factor influencing the disease.
Thank you. We got a question from Mary. Thank you for your wonderful presentation, for your wonderful research. What attracted you to FA and FA research?
Well, at the beginning, it was I was in Innsbruck, and Innsbruck was already a center for FA research. And I met my supervisor at the time, Sylvia Busch, that is working on FA since a long time. But after the PhD on FA, I went on working on this because I liked it, because it's interesting from a scientific point of view, and also because it's, I mean, FA patient, my FA patients are, I won't say brilliant, but yeah, they are determined and yeah, and it's a pleasure to work with them.
Okay, nice.
So maybe we'll move on because we have other presenters. You see that there's a lot of questions, so we'll
lots of questions. But thank you for the presentation.
Very hot topic, yeah.
I will stop my.
Yeah. We have to answer a few more questions.
I think we'll try to maybe get some information out to the ones that are asking the question, and we'll move on to the next speaker. Leslie, would you like to introduce the next speaker?
Okay. Okay, another question then. If you start taking iron due to deficiency, how frequently should you be tested?
So that, and I just put this in the chat. So that's really gonna depend on someone's personal situation, and this is definitely something that you should talk to your doctors about. And FARA is definitely noting that iron in FA is a topic that's of interest to the community. So we'll definitely consider how to bring this into other educational events that we put on. And thanks for the great questions. Leslie, do you want to introduce the next speaker at this point?
Oh, yeah, it's good. Okay, thank you, Lisbeth, for the presentation. And now we have Roderick Maas. He's from the Radboud University of Nijmegen. And he's did research about the peripheral nervous system and people in FA and how it affects people in FA. So Roderick, is on to you.
Yes, thank you, Leslie. Can you see my full screen slide or the small screen slide?
Yeah, we see the presenter.
Is this one better or worse?
Yes, there it is.
Good.
That's better, okay. Well, thank you very much for the introduction, Leslie. My name is Roderick Maas. I'm a neurologist, an ataxia investigator from the Netherlands. And it's a pleasure to showcase our peripheral nervous system imaging study that we're currently conducting. As baseline, visits of the study are currently ongoing. I do not have data yet to present, but instead the focus of this flash talk will be on the design and the rationale of the study, which is funded by FARA and Stichting Friedreich Ataxia Nederland. FA is characterized by mixed ataxia with involvement of the cerebellum, the spinal cord, and different parts of the peripheral nervous system. Damage to peripheral sensory nerves in the upper and lower limbs, as well as the cell bodies in the so-called dorsal root ganglia just outside the spinal cord, is a common feature in neuropathological studies in patients with FA. Their combination leads to a decrease in sensory input to the brain, decreased sensory feedback, which contributes to gait and balance difficulties in patients with FA, especially when the eyes are closed. This is a more complex figure illustrating the peripheral nervous system in somewhat more detail. It shows the pathway traveling from the skin to the peripheral nerve to the dorsal root ganglion, where the cell bodies of the sensory nerves are located, to the spinal cord, and finally upwards to the brain. It also illustrates that individual nerve fibers are bundled together in so-called fascicles, and that multiple nerve fascicles, together with blood vessels and connective tissues, make up the peripheral nerves in the arms and the legs. Our current knowledge on peripheral nervous system involvement in FA is largely based on autopsy studies and also on nerve biopsy studies. These studies allow comparisons between patients of different ages, different disease severities, but because of their cross-sectional design, do not address progression within individual patients with FA. Our study is the first longitudinal peripheral nervous system imaging study in patients with FA that focuses on both the peripheral nerves on the left side and the dorsal root ganglia on the right side. We use nerve ultrasound to measure cross-sectional areas of the two lower extremity nerves, two upper extremity nerves, and cervical nerve roots. In this image, we see a median nerve at a level of the wrist with the typical honeycomb appearance that defines a nerve architecture. In addition to the conventional nerve ultrasound, we use another technique that's called ultra-high frequency ultrasound, which allows a more detailed visualization of individual nerve fascicles with a very high resolution. We also perform nerve conduction studies of the same set of nerves that undergo imaging. So we have an idea about the structure of the nerves and the function of the nerves. For the deeper located dorsal root ganglia, we use advanced MRI techniques of the lower back and measure volumes of the individual ganglia at different levels, both sides, left and right. We obtain imaging data at a baseline visit and a one-year follow-up visit, and complement these imaging data with several questionnaires, so patient-reported outcome measures, several neurological tests, clinician-reported outcome measures, to evaluate the actual clinical relevance of imaging abnormalities in FA. These include like ataxia severity, quality of life, fatigue, activities of daily living, just to name a few. And finally, we will measure concentrations of neurofilament light chain in the blood, which reflects damage to large myelinated nerve fibers, and evaluate relationships with nerve sizes and dorsal root ganglion volumes. So taken together, we think our study will contribute to a better understanding and a visualization of the sensory neuropathy in patients with FA, fill a knowledge gap about the annual progression rate of peripheral nervous system degeneration in FA, and maybe hopefully yield imaging markers of disease severity and disease progression that may be applied in future therapeutic trials of patients with FA. With that, I'd like to thank you for your attention. Happy to take questions.
Thank you, Roderick. Are there any questions? Not yet. Maybe, no, not yet. Did you already research people with FA?
Whether I already research patients or whether similar studies have...
Yeah. No, you on this program.
Well, we are currently conducting the baseline visits of the study, and we have seen up to now 11 patients with FA.
Okay.
We aim to recruit 25 patients, which will make it the largest peripheral nervous system imaging study in patients with FA. So far, there have been 2 smaller nerve ultrasound studies from New Zealand and also from Italy. They evaluated 8 patients and 10 patients, so they had a smaller sample size. And I'm not aware of any published papers on dorsal root ganglion imaging yet in FA.
Okay, thank you. Yeah,
There's a question. There's a question from Elizabeth.
Yeah, from Ron Barzak. What are your observations regarding the health of the neurons of the dorsal root ganglia in these FA patients?
Sorry, can you repeat the first part of the question?
What are your observations regarding the health of the neurons of the dorsal root ganglia in these FA patients?
Yeah, well, we just started the study, and we're currently already segmenting part of the dorsal root ganglia of our patients, but we don't have comparisons yet with healthy controls, so we don't have a reference so far.
Okay, thank you.
But if we consider neuropathological studies in patients with FA, the dorsal root ganglia are quite prominently and quite early affected in the disease process. So I think we will observe smaller dorsal root ganglia in our study. And the question is if we see dynamics over time, if we see a decrease in volumes at 1 year follow-up visit compared with the baseline visit.
Okay, thank you. We got a good question from Michael. Do you think that with these methods, you will be able to detect the early differences in changes in lower and upper limbs?
Yeah, that's an excellent question. And up to now, we're not quite sure if it's fully degeneration, fully neurodevelopmental, or a combination of both aspects. We will consider upper limb nerves and lower limb nerves in the with the ultrasound aspects. Pilot data from the studies that I mentioned so far suggests that patients with FA might have enlarged nerves, especially in the proximal parts of the upper parts of the arms, and which might be associated with the severity of their ataxia. But these findings are very preliminary, only of 8 or 10 patients, and need to be replicated in larger sample sizes.
Okay, thank you. Another question from Mary. Wow, thank you. What are you most excited? What are you the most excited about for your research?
Well, I'm most excited about this being the first longitudinal peripheral nervous system imaging study with both ultrasound and MRI in patients with FA. So I'm very much looking forward to seeing the actual results, both from the baseline visits and even more the 1 year follow-up visits to see if we can provide imaging biomarkers of peripheral nervous system degeneration.
Okay, thank you. Last question. Do you have late onset patients in your study?
That's also a very good question. The patients we've seen so far, the 11 patients, are quite different. Most of them are, classic FA with early onset, but there are also a few patients with late onset or even very late onset FA. Yeah.
Okay. Well, thank you, Roderick, for your presentation. And now we're going to David.
All right, thank you, Leslie. And fantastic presentation so far. We're going to keep it going. Good things come in threes. So I'm David Castle, and we're going to hear from David Berryman and David Rufino Ramos. But first, just a quick bit about me. I live just outside of the city of Chicago and have a daughter who's 20 years old. She's a student at Illinois State University, and she has FA. And she was diagnosed probably about 6 or 7 years ago. So this is all really fascinating and important work, and just want to thank everybody who's on this call, both from the organizing, the presenting, and the questions that are being asked. But let's start with David Berryman, who's from the University of Florida, and I'll hand it over to you, David.
Awesome. Thank you for having me. So go back, please. Sorry about that. doesn't want to cooperate with me. I apologize.
We can see that if you just leave it. Yep, there you go. It looks good.
All right. So, well, my name is David Berryman, and my work asks the question, can we turn back the clock on repeat expansion? So like I said, my name is David. I was born and raised and continue to work in Florida, where I'm in the lab of Dr. Eric Wang at the University of Florida. Eric and the lab have been interested in repeat expansion disorders for a very long time, but I didn't know much about them until I had met Christian Moget, who joined the University of Florida Genetics and Genomics graduate program the same year as me. He and the now Dr. Chandra Trantham both live with FA, and they really opened my eyes to the disease. So when I had the opportunity to leverage my PI's expertise in repeat expansion disorders to the Friedreich ataxia field, I was really eager to do it. So ultimately, my work is founded on a fundamental principle of repeat expansion disorders like FA. Many of you are aware that with FA, you inherit GAA repeat expansions from your mother and your father. And then as you age, these repeats grow. And the severity of many of the FA symptoms are directly linked to how long the repeat you inherited, and then how quickly and long the repeat grows. So for example, someone who inherited 300 and 800 GAA repeats in the frataxin gene when they were born, these repeats could grow by dozens or hundreds of repeats by the time they were 20 or 30 years old. Depending on the tissue, where blood cells, these repeats might expand a little bit, but in the nerve cells that are responsible for ataxia, they can grow to be quite a lot, severely impacting cellular health. At the same time, paralleling this repeat growth, the severity of disease symptoms often gets quite a bit worse. So our idea to address this is relatively straightforward. What if we could stop the repeat from expanding and hopefully stop the clock on disease progression, and then investigate if there is a way to reverse this repeat expansion and cause repeat contraction? So our work really benefits from timely work being done in other similar repeat expansion diseases like Huntington's disease. Huntington's, like FA, is the result of a repeat expansion where the severity of symptoms is directly linked to repeat expansion over time. So they share the characteristic that the inherited repeat becomes longer as someone ages, and this parallels the progression of symptoms. Recently, however, it was shown that you can take certain protein systems, like the CRISPR-Cas9 system, that can cut DNA in specific places on the Huntington's disease repeat. And then when you cut this repeat, it actually can induce contraction. And there are also other DNA repair proteins that have been found to influence the rate and direction of repeat expansion instability. So our plan is to leverage what has been learned in Huntington's disease and other repeat expansion diseases, and then optimize it for FA, combining the cutting of DNA with the CRISPR-Cas9 system with a direct recruitment of repeat instability influencing factors like DNA repair proteins. And then if we're successful in inducing contractions, see if that could rescue cell health and alleviate some of the molecular symptoms of the disease. And with that, I'm happy to take any questions.
Thank you for that, David, and thanks for the presentation, but also thanks for describing how you came to the work of FA. That's a great story and inspiration to step into this work. I have one question, and just will remind folks to put some questions into the Q&A box. But so the very last statement that you made, you sort of said, if we could do this, then we could potentially achieve that. What does that mean for an average FA patient? And what could you imagine the results being if everything worked out the way you would like it to?
Well, I certainly won't try to get ahead of myself. There's quite a bit of work left to do. I think some of the work done in Huntington's is very promising, but what they've achieved with using the CRISPR-Cas9 sort of nicking approach to target repeats directly has been promising with a slightly different repeat motif. Huntington's is a repeat motif of a CTG compared to the GAAs that we know to be characteristic of Friedreich's ataxia. So we need to be careful to make sure that the mechanisms that have been successful with Huntington's will also be successful with Friedreich's ataxia. And so in the event that we do achieve any sort of decrease in the rates of expansion, or sort of a halting of the expansion itself, or if we do achieve any sort of contraction event, we'd like to assay that with molecular phenotypes like presence of reactive oxygen species, or the general transcription levels of the frataxin protein as well.
Great, thank you. And we did have a question come in. How can you determine if the treatment reaches the critical number of cells needed to achieve therapeutic benefit?
Yeah, that's a fantastic question. So most of this work is still on the basic science side. The ability to then vector and then deliver all of these gene therapy, or all of this sort of gene editing and therapies would be a different animal altogether. One that I imagine David, David Ramos might address himself. But yeah, the vectoring it is a separate and equally, if not more difficult issue.
Could I ask a follow-up question? And maybe I just missed this in your presentation. So are you doing this like in dishes of FA cells? Are you doing it in animal models?
Yeah, I've been doing this with patient-derived iPSC, so patient-derived stem cells.
So those are like cells that you have in front of you in a dish or a flask, and it's very different to get the editing into someone's cells when they're in their body.
Yeah, exactly. Thank you for the clarification. This is something of a proof of concept. The idea that if we can prove that contraction or the halting of expansion can be achieved, then we can move in a more simple model like cells in a dish that we have in an incubator. We can move forward with trying to address more complicated issues like how to deliver that therapy systemically and to actually achieve a therapeutic result.
Great. I think we have time for one or two more quick questions. What cell systems are you currently using? Any FA cells like fibroblasts?
I do have some FA fibroblasts, but I have found the most success with Friedreich's ataxia patient-derived iPSCs, and those have been fantastic. There's been some discussion about when we plan on differentiating these into more mature neuronal subtypes, and see if sort of the different transcriptional profiles and how the frataxin protein is expressed in slightly different cell types may affect instability. But right now we're still working with base stem cells.
Great. How are we doing on time? One more, or should we move on to David?
Yeah, let's ask the question on about cutting out. Maybe you can clarify, because your plan is not to cut out the repeat, but you're doing something different, and what are you doing is different from cutting out the repeats, and is it safer? You can spend a minute on that.
Yeah, absolutely. So the big concern, if we were just to take these pairs of scissors here and cut out massively all of the GAA repeats that are causative of the disease, is we'd be really concerned about off-target effects of the Cas9 protein and potentially deleterious effects on other genes that might be vital for cell health and your health. So what we do actually is, instead of cutting across the DNA in both strands, we take a adapted CRISPR-Cas9 system that will only cut one strand and then target a specialized repair cascade that then causes sort of incremental contractions. So you have a little bit more control, and there's less chance for a abnormal deleterious effect to happen in the general genome.
Great, thanks so much. There were a couple of questions we didn't have time to get to. David, I don't know if you can see the questions in there, but maybe you could answer those in the chat. But let's move to David Rufino Ramos, the third David in the group, who's from the Massachusetts General Hospital as well as the Harvard Medical School. And with that, I will hand it over to you, David.
Do you hear me?
Yes, we can hear you. Go ahead.
Okay. Yeah. Okay. Hello, everyone. Thank you so much for the invitation to be here to show some of my thoughts and the work that I've been doing in Mass General Hospital. I'm David. I'm originally from Portugal, and I came to do a postdoc in Boston, USA. And I've been working in repeat disorders for all my scientific life, but I just started working with Friedreich ataxia about 1 year ago. So we are mainly a genome editing lab. So we work in the protein engineering side to creating tools that can hopefully apply these tools to treat certain disease. And we got very interested in tackle Friedreich ataxia disease and tackle the root cause of that. So we are targeting the DNA, and hopefully our two major goals are to contract the GAA repeat or to replace to a normal length of the GAA. And for that, we want to leverage the genome editing tools that we've been developing in the lab that are basically proteins that we keep optimizing to expand the accessibility of the genome. So before we were a bit restricted in the genome accessibility, then we create enzymes that would have much more accessibility to the genome, but they were less safe. And now we create a much broader number of enzymes that are much more safer and also have the accessibility to different regions of the genome that were not available until now. So this offers enormous possibilities of tackling the Friedreich ataxia disorder at the DNA level in a much broad range. And of course, we can apply these proteins that we have been engineering to multiple genome editing tools that have been in the field for a long time. Probably you guys heard about base editing and prime editing, but very interestingly, we very recently create a new genome editing tool that is called Click Editor, and it has Cas9 nickase, that is these enzymes that we have been evolving, and that when associated with a guide RNA, it directs this tool to a specific region of the DNA. And associated with this, it has a H2H endonuclease, that is this small orange portion here, that just allow us to recruit a template. And this template is a portion of a DNA that you can basically tailor whatever edit we want to do. So we can give the instructions to encode in this template whatever we want to do. And we can use this either to delete the GAAs or to replace to a normal size GAAs. And for that, we're going to use a polymerase that is here highlighted in pink. And basically, this polymerase would write through this template and would create an edit that can be hopefully incorporated in the genome, and then would lead to the rescue, hopefully to the rescue of the pathologic repeat. And when we try this for the first time in cells, so in a dish, in a plate, we do see some promising results because we saw that this concept is working and is working for when applied for the Friedreich ataxia context. But we did think that we could have done much more to improve this system. And this is what we decided to do. we decided to take lots of engineering campaigns to improve each component of the system and also the template in order to create a tool that is just more efficient and also comes with more safety. And this is the results that we got. We got a much more potent tool that is much more efficient than the first generation tool that we created. So at this point, we are finalizing these small refinements on the tool and on the template, and then we want to take it to more complex, to apply this tool to more complex systems, such as patient fibroblasts and iPSCs. And for the next part of my project, I'm also going to focus on the delivery aspect. And in this delivery aspect, what we want to do is to give this tool in a very transient manner, so the tool doesn't stay there too long to minimize the possibility of any safety issue. So we're going to deliver this in a transient manner, and we're going to leverage all the expertise that I got during my PhD on non-viral delivery vectors. So we're going to just do non-viral delivery to transport all of this engineering tool, and hopefully we can get to different cell types, different tissues, and ultimately get to in vivo models. I'm very happy to be here and to answer any question that that I know. Thank you.
That was great, David, and thank you for the presentation. And I do believe that you tried to put that in the most layman terms, but even I am sort of wowed by the science, and so might rely on some of our FARA colleagues to maybe to start us off with a question that might sound like you know what's going on with this.
Maybe I can ask this one question about the last part that you were talking about. You were saying that you are trying to develop new delivery methods, and maybe can you tell us a little bit about what is currently being developed and what the difference is between what you're trying to develop and technologies that are already out there, like viral delivery for gene therapy, and why you need these special delivery methods that are different from the methods that are developed for gene therapy for this tool?
Yeah, thank you. This is a very good question. First of all, I'm sorry to put some data or to put a little bit more complexity on this. After I was seeing the other talks, I was like, okay, maybe I should have put it in a more layer format, but I would like just to give some graphs and some data just to get people excited also about the research that we are doing. Yeah, regarding the delivery aspect, so we have basically two main delivery approaches that we can take: viral delivery and non-viral delivery. And we have both been applying for other kind of disease in the clinic. The main concern of using viral delivery for genome editing tools is that these genome editing tools would stay probably longer, or they would stay longer in the cells, and this longer expression in the cells can potentially create some safety issues. So even if the tool is very safe, the fact that the tool stays longer there and being always there can create safety issues. So the genome editing field is moving towards to deliver these tools for a more transient way, which is basically you take advantage of the fact that we are targeting the DNA, and all the modifications that we are doing gonna be there forever, gonna stay in the DNA, and then you want this tool to disappear. You don't want this tool to be there. And we can get this by using non-viral delivery methods, and we can use lipid nanoparticles, or we can use more endogenous particles that are called extracellular vesicles. Or we can do some kind of combination between them. And basically, we can leverage both properties. And the idea is that we can create an efficient tool that, one, is able to package and deliver everything that we want to package. This is a engineering a system that is big, that is a system that have multiple components, and we need to be able to put this system all in one particle. And then we also want to engineer this particle to be scalable and also to be able to target different organs. Hopefully, we want to target all the cells, but this is a delivery issue that is not easy to solve. So basically, the targetability of the issue would bring how far we can take this tool, this concept, to therapy and to patients also.
Thanks. That's very helpful. And I think Kellen or Liz, there's a bunch of questions that have come through, and I know we're short on time. And I wonder if there's one that you can identify that might be a good one to hit before hopping off.
Yeah, I mean, we can ask about the question of where if you were able to rescue after you use this tool and you edit the repeats, are you able to rescue the frataxin level?
Yeah, that's a very good question. Very good point. We didn't evaluate the protein level yet. We are working at the DNA level, and we were trying to solve the major problem that we detect in the first part, that is trying to get these editing levels much higher. But we do need to do that, and we need to see that the protein level can be rescued by doing that. Yeah.
And then there's a question about the safety. If somebody is asking you about off targets, first of all, can you express what off targets are and if this tool has these off targets effects?
Yeah. Yeah. So when we engineer for a protein that is necessarily more efficient, we are always concerned of having a good ratio between efficiency and safety. And when we talk about safety, we talk about off targets. We talk about one of the things that we talk about is about off targets. And we talk about off targets is basically the ability of these tools to be able to edit other regions of the genome that we do not want them to edit, that are unintended, that create unintended edits. So basically, ideally, we want to create a tool that can be very efficient editing the precise site that we want to treat, but that can have very low minimally possibility of having off targets. Moreover, what we do after is, even we create a tool like that, we after want to reduce even more this possibility by just giving this tool in a transient manner. So we are even reducing more this probability of creating off targets. Yeah.
Maybe the last one, I will ask what Mary wants to know. What is the next step? Where are you going next with this research?
There's a lot of many steps to go further. So we are still doing some optimizations in the tool. We want to create a very good tool, a very robust tool that allow us to move to more complex systems. So as someone was saying before, we are doing this in cells, in a petri dish. We are just doing this and try to see if this is working or not, but this is not enough to say something about for a patient, right? So we take advantage of using these petri dishes to develop a very good tool that is very efficient and very safe. And then we want to move to patient cells and different kinds of patient cells, and also to more complex organs and ideally animal models. That's our final goal, where we also need to play with the delivery system. Yeah.
Great. All right. I think we reached the end. Thank you to the speakers today. Everybody did an excellent job. Thank you also, Leslie and David, for helping us moderate the session. And thank you all for listening. We hope you'll consider participating in research. There's a lot of ways you can participate. Look at the FARA website, check out our announcement on social media. And if you have participated in research in the past, thank you so very much. And I hope you enjoy learning about some of the research that FARA funds, and we'll do this again next year. Take care, everybody.